All right, let's go ahead and start by answering this bell ringer. The question asks, how might an amino acid change at a site distant from the active site of the enzyme alter the enzyme's substrate specificity? Well, the term specificity refers to the fact that an enzyme has a very specific substrate that it's going to bind to. It won't bind to a whole array of substrates. For example, like lactase is only going to bind to lactose, a type of sugar. It's not going to bind to glucose. It's not going to bind to fructose. It's only going to bind to one specific sugar. So that's what the word specificity means. If you read all the answer choices, the only one that really makes sense is C, by changing the shape of the protein. So that is the answer for this bell ringer for today. In this lecture, what we're going to do is talk about the innards of photosynthesis, like what actually happens during photosynthesis. We learned about in regular bio that photosynthesis is when a plant brings in sunlight or takes in sunlight, brings in some carbon dioxide, and then somehow forms glucose from this entire thing. And that's a very, very oversimplified definition of what photosynthesis is. So what we're going to do today is talk about how all of that actually works and how all of that actually happens. Now, remember from regular bio that photosynthesis is the conversion of light energy to chemical energy. That light energy can be from the sun. It could also be from grow light. So I know like a lot of people like if you want to start a garden in Minnesota, typically you would have to start your garden indoors, probably around April. Now, in April in Minnesota, the weather is very unpredictable. It could be very nice and sunny. It could be snowing. So it's very unpredictable. So a lot of people start with a grow light and then transition their plants outdoors later. But that either way, the energy source is going to be a light source. And the organisms that use photosynthesis are referred to as autotrophs. Troph levels refer to like how an organism gets their food. So an autotroph is going to be an organism that can produce their own food from substances in their surroundings. So plants will be able to get their own food from the sunlight plus the carbon dioxide they are able to bring in. A heterotroph is unable to make their own food and basically needs to consume autotroph or other heterotrophs in order to survive. Now, for the evolution of photosynthesis, the photosynthesis has been around for millions and millions of years. It's first evolved in prokaryotic organisms. We learned about the endosymbiotic theory back in Unit 2 and how chlorophyll is thought to be an old prokaryotic organism that was basically taken in by a eukaryotic cell and now lives in this symbiotic relationship within the eukaryotic cells. And so it's been around for a long time. Chlorophyll has been it's a very ancient organelle. And cyanobacteria is one of the first organisms to be thought to have that organelle. It brought oxygen to Earth's atmosphere a long time ago, billions of years ago, and it led to eukaryotic photosynthesis. All right, so when we talk about photosynthesis, we're not going to be referring to like just the chlorophyll. We're going to be talking about plants in general. More specifically, what part of the plant is actually doing the photosynthesis? It's going to be the leaves. We know chlorophyll is green, and we know chlorophyll is the site of photosynthesis. So what's the greenest part of a plant? It's going to be the leaves, and that's where the photosynthesis is actually taking place. But if we want to break that down even further, where exactly in the leaf is the photosynthesis occurring? Well, we have this cross section of a leaf. If we were to take a cut out of a leaf and kind of zoom in, some leaves have this like cuticle thing on the top. That cuticle is basically like this waxy layer, and that's just to help the plant be protected from loss of moisture, some other things that might try to affect the surface of it, maybe like intense UV rays. Not all plants have a thick cuticle layer, but most plants will have at least some form of cuticle layer on top. And again, it's going to be like this waxy texture. If you've ever taken a leaf from a tree, there's clearly a texture difference between the top of the leaf and the bottom of the leaf. And the top usually has some kind of cuticle layer. Right underneath that is the upper epidermis. This is where you can have cells that have very large vacuoles. Not a lot of photosynthesis happening here in the upper epidermis, is just basically a protective layer. And then underneath that, you're going to have basically this whole layer is called the mesophyll cells. That's the inside leaf tissue. This is where all the chlorophyll is going to be. And each one of these ovals that you see here, these light green ovals, that's supposed to represent the chloroplast, and that's where the cell will actually do photosynthesis. Now, that's the first part of photosynthesis, is the chloroplast and the chlorophyll inside of it. The sunlight does need to get in here, so sunlight is one part, but the other part is the carbon dioxide. Where does the carbon dioxide come into play? How does it get into the cells? Well, on the bottom of a leaf, if you've ever taken a leaf and put it under just a standard light microscope, you can actually see these openings called stoma. A single one is called a stoma. Multiple ones are called stomata. These are specialized openings that are comprised of two guard cells. So here's one guard cell here. Here's another guard cell here. They kind of form like, I kind of think they look like elbow noodles almost. And when they swell up with water, they will actually close this gap here. So the stoma will actually close. It's literally a pore on the bottom of a leaf. And when there's less water on the inside of these guard cells, they'll relax and open up, and the stomata will actually open up and allow gases to diffuse in and out. So this pore is extremely important because it's basically the respiration of the tree. This is where the tree or the plant will be able to bring in gases, also release gases. Things that go in are going to be carbon dioxide, sometimes water vapor if it's a really, really humid outside. If it's like 95, 100% humidity, then sometimes you might have a little bit of water vapor, but usually not. And this is also the site where oxygen comes out. Now, none of this happens through like a respiratory system. This is all just simple diffusion. And another thing to keep in mind is that water vapor can also leak out of the stomata. So that's very important to understand, is that we'll talk about that later. How do plants prevent a loss of water if water can easily escape through the stomata? So just keep that in the back burner. We'll mention that later, how plants can manage loss of water like that. All right, and we're going to zoom in on one of those chloroplasts and kind of take a look at the inside of the chloroplast. We did talk a little bit about the chloroplast in Unit 2 and how it's an energy organelle. Just to kind of review the anatomy of the chloroplast, you have the outer membrane, and then you'll have an inner membrane space right between the outer membrane and the inner membrane. And then inside of the inner membrane, you have this space, and this space is called the stroma. So don't get the stroma confused with the stoma or the stomata. So very confusing. It can get very confusing if you don't know the difference between the three. Stoma is a pore. Stomata is plural of that, so it's multiple pores. And then stroma with an R is the space inside of the inner membrane of a chloroplast. Another thing that you see on the inside of the chloroplast are these little discs, and these discs are called thylakoids. And this is actually where the reaction for photosynthesis is going to be taking place. The thylakoid has a membrane on itself. So I'm putting my cursor right across one of the membranes of the thylakoid. And so the thylakoid membrane has two parts. It has the stroma on the outside, and on the inside of the thylakoid membrane, it's got the lumen. So those are the two areas that it's separating, the stroma and then the lumen on the inside. All right. One other vocabulary term that we'll throw in here is that these thylakoids, they are often found in stacks just like this, and these stacks, they are called a granium. So just to kind of maximize surface area, maximize the reactivity of the photosynthesis, to maximize how much photosynthesis actually takes place. So if we remember from regular biology, maybe your biology teacher had you memorize this equation for photosynthesis, where you have 6 carbon dioxides plus 6 water molecules, and then if you included light energy, what you ended up with was a glucose molecule, C6H12O6, plus 6 oxygen molecules as a byproduct. Now that's basically what we're going to see here. We're going to see an input of all of these things, but we're going to actually break this down a little bit to talk about how all of this comes into play. Where do we see all of these things jumping in? So we're going to talk about photosynthesis in two stages. The first stage is going to be the light dependent reactions. And the light dependent reactions occur inside of the chloroplast, but specifically across the membrane of the thylakoids. So if you remember, the inside of the thylakoid is the lumen, the outside of the thylakoid is the stroma, and the difference between the two across that membrane is where that light reaction is going to occur. And this is what I call the photo part of photosynthesis, because light is absolutely necessary in order for those reactions to happen. And that's kind of like the first part. You do need water in that step, and you do need light energies. This is where those two things come into play. Water and light energy, those two things, those are going to be the things that are definitely necessary for the light dependent reactions, and they're responsible for kind of kicking off the light dependent reactions. The products of those reactions, once we've completed all of those reactions, those products will move on outside of the thylakoids into the stroma, and then undergo this process called the Calvin cycle. Now, the Calvin cycle sometimes also has other names. Some textbooks call it the dark reactions. I don't really like that term, the dark reactions, because it's basically assuming that there's no light available, which is not necessarily true. I like to call them the light independent reactions, which is what most textbooks will call it. The light independent reactions is more accurate because these reactions can occur during the daytime where there is light. It's just the light is not necessary. There's no light being involved here because there's no more pigment absorbing light anymore outside of the thylakoids. So this is the synthesis part. This is where actually we're taking carbon dioxide and we will be then combining it with whatever products we got from the photo part of photosynthesis or the light dependent reactions, and we'll be making glucose from this whole thing. The other thing I do want to mention before we move on, the oxygen is a byproduct of the light dependent reactions. So water, light energy, and oxygen are all part of the light dependent reactions. Carbon dioxide and glucose will eventually be part of the light independent reactions. All right. I know it's a lot of information to cover all at once in this slide. We will be going through this whole process meticulously and a little bit slower just to make sure that we understand where everything falls into place. So now we're going to mention a little bit about wavelength and how that relates to photosynthetic pigments. So we're going to jump into physics here a little bit. But photosynthetic pigments absorb light energy of different wavelengths. So I've mentioned chlorophyll before. Chlorophyll is the main component, the thing that's going to be driving a lot of the photosynthesis in the beginning. There are also pigments called carotenoids. Things that have a lot of carotenoids, like carrots obviously have a lot of carotenoids. Pumpkins have a lot of carotenoids. Things are usually like orange. And we know chlorophyll is usually like a green color. And these pigments are actually embedded inside of the thylakoids, and they're very good at absorbing a specific wavelength of light. So let's talk a little bit about that and what that means. So when we understand light, light is basically a movement of the electromagnetic spectrum. It's a wave that travels through the electromagnetic spectrum, and it's made out of particles of energy called photons. And as photons move at a specific wavelength or a specific frequency, they will produce a wavelength that can then be attributed to one of these different types of waves. If you have really long waves, those are going to be like FM and AM radio waves. Sometimes like the old TV, TV used to be broadcast over analog, and you could pick it up with an antenna. And so you could actually get TV waves. Microwaves that we use in like our microwave ovens, or even our phones, our smartphones use microwaves. Those are all types of wavelengths that we see. On the higher end, we see really like dangerous gamma rays that we see in radiation. X-rays, those are also pretty dangerous, and ultraviolet. All of these are very capable of causing things like cancer, and they're very high energy waves. And in between all of these waves, there's a very narrow band, and that band includes all visible light. And that actually is a measurement. You can actually see the measurements of all the wavelengths of light. Anywhere from 700 nanometers to 400 nanometers is basically what humans can see. There are animals that can see more, like there are like reptiles that can see infrared. There are a couple of organisms that can see ultraviolet light, but for the sake of this PowerPoint, we're going to stick to basically what can the plants see, like what are the pigments able to absorb which wavelength of light. When I refer to wavelength, of course, it's the distance from the crest of one wave to another wave. And so basically, when I say 700 nanometers, that means that's the width of one wave. It's very, very small, and it's traveling through the electromagnetic spectrum, and those photons are hitting your eyes. And each one, if you're looking at the color red, for instance, that's going to be 700 nanometers in width. And the big ones that we like to look at are 380 nanometers and 750 nanometers. Those are going to be that's the big range here for all colors of light. I'd also like to mention that shorter waves will have a higher energy. So if you look at all of these different colors, they are not equal in terms of the amount of energy that they can capture. The purple light, for instance, or violet, I guess, that's going to be the highest amount of energy in terms of visible light. The long wavelengths are going to be lower energy, like the red light here, that's going to be the lowest amount of energy. So when light interacts with matter, it can either be reflected, transmitted, or it can be absorbed. And the absorption part is actually going to be the most important when we talk about pigments, because pigments are able to absorb pretty much all of the colors on the spectrum in terms of visible light, except they don't absorb one specific color, and that color happens to be green. Leaves are green because the chlorophyll will absorb every single color, except it will reflect back green. So whatever it's reflecting back is what we see, and that just happens to be the least efficient wavelength of light that plants can absorb. So let's talk just about what kind of pigments that we see, and we can actually see what wavelength they can absorb. Chlorophyll A is one of the more primary pigments. It's involved in the light reactions. It's usually like a blue-green color in terms of like when you look at it, the pigment itself. And what that means is that it's absorbing every single color except for like this bluish-greenish hue. Chlorophyll B is an accessory pigment, and it's kind of like this yellow-green. So it kind of evens out the chlorophyll A and makes plants look a little bit more green. And there's also carotenoids, and these help broaden the spectrum of colors that drive photosynthesis. It's a yellow-orange pigment, and it's used as photoprotection. So carotenoids absorb and dissipate excessive light energy that could damage chlorophyll or interact with oxygen. So these are just 3 examples of pigments that we see. I'm not expecting you to like memorize these pigments or know what their ranges are for what they absorb in terms of wavelengths. All I need you to know is that there are different types of pigments, and then basically they absorb different colors. All right, so now we've learned a little bit about the pigments. We're going to reference them in this diagram, which does look a little bit scary at first. And this is actually going to be our kickoff to photosynthesis. We're going to go ahead and start off photosynthesis looking at this overview. So I've got a couple of animations that I'm going to show you on this slide in particular to kind of explain this whole process. There's a lot going on in this rectangle, but we're going to walk through it step by step. The first thing we want to note here is where are the light reactions occurring? Okay, so we're going to go back a couple slides here. And remember from this slide, I said the photo part is the light-dependent reactions, which is what we're going to talk about right now. And that is occurring inside of the chloroplast right here. Specifically, light activation of one of the pigments, one of the chlorophyll pigments. Okay, and that's occurring across the thylakoid membrane. So right here, that little cross section, that rectangle that I'm pointing to with my cursor, that's supposed to represent this rectangle right here. You can see the phospholipid bilayer behind us right here, kind of grayed out. That is supposed to represent the thylakoid membrane across here. Above it is the stroma, and below it is the lumen. This is the inside of the thylakoid. All right? So what we're going to do is kick off this reaction. We need two things in order to kick off all of photosynthesis. You need water and you need light. So we're going to go ahead and start with our water molecule. And what's going to happen is that water is going to approach this green blob here. And this green blob is actually a series of proteins. It's like a cluster of proteins. And we call this cluster of proteins photosystem 2, PS2, or photosystem 2. Okay, and it's going to move up here. So as water approaches photosystem 2, it actually got close to this pigment right here, P680. And this pigment absorbs a wavelength of 680 nanometers. I'm going to go back a couple slides here. 680 nanometers is probably somewhere in this range or so. It's probably like red or so. It's going to absorb that light. So remember, it's absorbing that color, so it's reflecting all the other colors as well. So water is going to approach photosystem 2, and then we're going to have light activating this whole reaction. So light's going to come in. There's my little lightning bolt to represent light. And as light hits that pigment, what that pigment is going to do, what this whole cluster of proteins is going to do, photosystem 2, it's actually going to separate our water. Now, when you separate water, you're breaking covalent bonds. And when you break covalent bonds, you're actually going to have to separate electrons. So what's going to happen is that we're going to break apart that water, and we took the electron from that whole reaction. You saw the 2 hydrogens float away, and we saw 1 oxygen float away. Okay? Now, eventually we'll have to do this twice. We'll have to do this with 2 water molecules, because when oxygen diffuses out of the cell, it is going to have to be in the form of O2. Because oxygen is a diatomic element. When it forms a gas, it's always O2. It's never just by itself. So we'll do this again with another water molecule, and eventually that oxygen will pair up with another oxygen to form O2 and then diffuse out of the cell. But right now we're going to focus on that electron that just got moved. So we broke apart that water, and that electron is now in an excited state. So you can kind of see we lifted it up a little bit to represent that it's in an excited state. And we're actually going to transport that electron through a series of peripheral and integral proteins within the thylakoid. So it's going to go through our first peripheral protein called plastoquinone, and then it's going to go through another one called cytochrome. But as it's going through all of these peripheral and then the integral proteins, it is going to be losing energy. As it goes through cytochrome, a lot of that excited state of the electron, it's passing off its energy in order to pump a proton through the membrane. So you can see that we took the hydrogen from one side of the membrane, where it is in the stroma, and we put it inside the thylakoid. So every single time we move that electron through, I'm going to show you again, we move the electron through, we also pumped a hydrogen through this enzyme called cytochrome. And that's going to be important later, because we're going to need to show that we're actually building up a large concentration of protons here in the bottom near the inside of the thylakoid, inside the lumen. All right, but we're going to continue talking about this one electron here, because he's kind of important. He's going to go through another peripheral protein called plastocyanin, and then he's going to approach another cluster of proteins called photosystem 1. And once again, we're going to have light energy activate that chlorophyll, and then it's going to excite that electron. Now, this electron's in an excited state. We're going to pass it on to the next thing. We're going to pass it on through another peripheral protein called NADP reductase. And then we're going to take one of those protons that's inside of the stroma plus that electron, and we're going to combine those two and attach it to a molecule called NADP+. NADP+ is an electron carrier. I'll talk a little bit about that in just a little bit, but it's positively charged, and then we combine it with that electron and another proton, and then that proton gets attached to NADP plus to form NADPH. So a lot of stuff going on here. We've got this proton that's going to combine again with that electron. The proton is that hydrogen ion, remember, and it's going to combine with NADP plus to form NADPH. Okay. Last step is that we've taken care of this hydrogen here, but there's a whole bunch of hydrogens down here. Okay, and we don't want that because that's going to lead to a very acidic environment. Remember, pH is the measurement of hydrogen ions. The more hydrogens you have, the lower your pH, and it's going to end up with a very acidic environment. So we got to do something with all of these hydrogens that are building up. By the way, where did all of these hydrogens come from? Two sources. These two hydrogens that we saw earlier came from the splitting of water. When we split water, we ended up with some hydrogens here. The second source of hydrogens was when we lowered the energy of that electron through cytochrome. We had to move a hydrogen through this this integral protein, the cytochrome. So we did that intentionally to build up a whole bunch of protons here, these hydrogens. So why did we do that? Why did we build up a whole bunch of these? Well, this is called the concentration gradient, or chemiosmosis is the term, where it's basically kind of like osmosis, but with a chemical, and that chemical happens to be all these hydrogen ions. We can then utilize this giant gradient between having a bunch of hydrogens here and having no hydrogens here to push them through, kind of like osmosis, through a specific enzyme called ATP synthase. ATP synthase is an extremely important enzyme. We'll talk more about it when we get to cellular respiration, but basically what ATP synthase does, it synthesizes ATP. The way it does that is it takes a proton, it pumps it through ATP synthase, and that energy alone will allow ADP plus a phosphate to turn back into ATP. Okay? And we'll talk a little bit about that later. But one thing I do want to talk about first, and we'll come back to that diagram in a second, but I do want to talk about this guy over here that we kind of looked over, and that was NADPH. What exactly is NADPH? Well, NADPH is an electron carrier. You can kind of compare it to like a pickup truck. It's basically loading on that electron in the back of its bed, and when it's not carrying an electron, it is written as NADP+, so it's got a positive charge to it. When it is carrying that electron, it's storing that electron in the bond between NADP and the hydrogen ion. Now, it's very important to understand that there are different types of energy carriers or electron carriers. They're not to be confused with another electron carrier that we will learn about later in cellular respiration, and that one is called NADH. So it's very confusing. We have NADH and we have NADPH. I like to think of as the P as the one standing for, like, that's the one I see in plants, that's the one I see in photosynthesis. So NADPH, that's the way I remember it. And again, this is just basically kind of like a truck. It's carrying electrons. We're going to need this thing, this NADPH, for the light independent reactions. So this is basically all of the light reactions. This is all the steps necessary that require light. And the products of this are, well, we have NADPH. We were able to get some energy in the form of ATP. And we also had our byproduct of the oxygen, which is not really important. And that's basically, it's a waste product for the plant. We don't really care about that. But in us humans, we do care about that because that's actually where we get all of our oxygen from. So those are the big things, the big carrying points. Like, what do we take away from this? We got, here's our summary. We converted solar energy to chemical energy. Chemical energy produced in two forms. We did get some NADPH. It's the electron carrier. You can kind of think of that as like the battery, okay? By itself, it's pretty useless, but it does hold energy that we will be able to use later. And then we have ATP, which is like a true power source. And then we also split some water. Splitting water provided the source of electrons and protons for the this whole reaction. It released oxygen gas as a byproduct. Light absorbed by chlorophyll drives the transfer of electrons and hydrogen ions from water to an electron acceptor called NADP+. NADP+ is then reduced into NADPH. Don't forget about oxidation and reduction, those two terms. NADPH is the reduced form, and then we generated some ATP. So again, two products and one byproduct. That's a big takeaway. Our two products, ATP, NADPH, one byproduct, oxygen gas. We're going to just take a look at this animation one more time. We'll zoom back to the very beginning, kind of see this whole process again. I'll walk through it one more time. So water comes up to photosystem 2. Light splits water. Then we pull an electron from water. The water molecule separates into 2 hydrogen ions and an oxygen gas. That electron gets passed through cytochrome, which pumps yet another proton through the thylakoid membrane. The electron keeps on getting passed, and it goes into another set of enzymes, and that whole thing is called photosystem 1. Light activates that molecule again, excites the electron. It gets passed to something called NADP reductase, which its whole purpose is to take that electron and pass it off to NADP+ along with an extra proton. And so these are the big products. We got oxygen, we got NADPH, and ATP. Now, last thing we're going to note about the light reactions is that there are some cases in plants in which instead of the electron being accepted by NADPH, sometimes we don't make NADPH. Sometimes we take that electron and we loop it back to the first part of this electron transport chain. So we have the electron kind of zooming back here to the first part instead of going to NADPH way over here. And so that occurs between photosystem 2 and photosystem 1. And that electron is basically recycled and reusing it over and over again. And this just helps certain types of plants make more ATP. We'll learn more about that in this PowerPoint. These types of plants are commonly referred to as C4 plants. We may not need as much NADPH, and instead we'll be making more ATP because we'll have more protons coming through. Remember, when that electron came back, when it came back over here, and we recycled it going back through cytochrome here, we pumped more of those protons through the membrane. So we have a proton here, we have a proton here. And that electron came back and helped move those protons from here and here to the inside of the membrane. So eventually what you're going to have is a whole bunch of hydrogen ions, a whole bunch of these protons floating around on the inside. And eventually they will go through ATP synthase. When ATP synthase gets hydrogens going through, with every single one proton, we get ATP being made. All right, so that concludes our light-dependent reactions. In the next part, we're going to talk about the light-independent reactions, where we're moving outside of the thylakoid, but we're still inside of the chloroplast. So we're no longer reliant on this whole mechanism or this whole membrane. We're going to be outside here in the stroma, and we're going to do the next part. Remember, now we've got ATP floating around in the stroma. We also have, if we are talking about a different type of plant, like a normal plant, we've got NADPH floating around in the stroma as well. So those two things are going to be carried forward along with carbon dioxide. That is going to be another reactant. Those two things are going to carry forward into the next part, into the light independent reactions and the Calvin cycle.